everybody. Thank you for joining us to the webinar on innovative solutions for cGMP-grade human mesenchymal cell culture. My name is David Fiorentini. I am the VP for Scientific Affairs and Head of R&D in Biological Industries. If you have any questions, you may ask during the presentation using the chat module, and our researchers will answer you online. Human mesenchymal stem cells are multipotent adult stem cells. They are adherent fibroblasts-like with spindle-shaped phenotype. We may isolate the cells from various sources like bone marrow, adipose tissue, umbilical cord, amniotic fluid, placenta, and dental pulp. We may expand the cells in vitro, and we may use them for various applications like basic biology, cancer research, drug discovery, and cell-based therapy. These cells may be differentiated into cartilage, bone, and fat, as well as into other cell kinds like neurons, hepatocytes, and endothelial cells. Human mesenchymal stem cells have become of interest for clinical applications due to accessible source of cells with no ethical issues, expansion capability, multipotency. They can be differentiated into various cell types, capacity for homing and engraftment into injured tissue, secretion of bioactive molecules for recovery of injured tissue, and inhibition of inflammation, and they are immunoprivileged with low immune response, and we may use them in autologous or allogeneic kind of treatment. According to clinicaltrials.gov, in May 2015, there were more than 500 clinical trials done all over the world using mesenchymal stem cells. So having all this in mind, we had a mission. And the mission was to develop an efficient, versatile, and consistent xeno-free culture system for human mesenchymal stem cells towards clinical applications. And this system should include culture media and auxiliary solutions for isolation and expansion of the cell and for differentiation. And this system should be suitable for cells from a variety of tissue sources. In the past, we used to expand cells using serum, and then later on we switched to serum-free culture media. But today, we would like to work with serum-free and xeno-free culture system, or even with animal components-free system, as this system is regulatory friendly. So what are the advantages of chemically defined xeno-free culture medium for human mesenchymal stem cells? It contains no non-human ingredients, what we call xeno-free. It is defined formulation with lot-to-lot consistency. The product is safe, reduce contagious health risks like viral agents, and limit the immunological reactions once cells are transplanted. And as so, the medium is accepted for clinical applications. You have to remember that the more defined your culture system, the more controlled and consistent cell culture you will achieve. As we started working with mesenchymal stem cells, we noticed that Foetal Bovine Serum actually has no advantage for the expansion of human mesenchymal stem cells. As you can see here, two versions of serum-free, xeno-free media in comparison to serum-containing medium, and you can see the difference in the number of cells as well as in the photos here, the density of the culture. This is serum-containing medium, and in this case we use xeno-free, serum-free culture medium. And you can see the difference in the density of the culture. The first two products that we developed as part of the system are the medium MSC NutriStem xeno-free. It is serum-free, xeno-free medium, specially developed for the expansion of human mesenchymal stem cells. The medium composed of two parts. One part is the basal medium, and the other part is the supplement mix that we keep frozen. When we add the supplement mix to the basal medium, we get the complete ready-to-use medium. And the second product that we developed is the MSC Attachment Solution. It is a xeno-free reagent for the attachment of human mesenchymal stem cells in serum-free, xeno-free culture system, and it is based on human plasma fibronectin. In 2013, most of the clinical trials done with human mesenchymal stem cells were with cells from bone marrow, umbilical cord, adipose tissue, and placenta. So we chose these tissues to start working with for the isolation of human mesenchymal stem cells using our serum-free, xeno-free culture system. And I would like to share some of the isolation data with you. The first tissue that we try to isolate cells from is the bone marrow. And in this case, even in the isolation step, we do not use any human AB serum into the medium. Sometimes with certain tissue types, we recommend to use human AB serum, 2% human AB serum, just for the isolation step. But in this case of bone marrow, there is no need to use human AB serum for the isolation step, and still you can see that you can get much higher number of cells using MSC NutriStem and MSC Attachment Solution in comparison to serum-containing medium. And when we did the immunophenotyping, you can see that around 99% of the cells express the MSC markers, and only low number of cells express the hematopoietic markers in comparison to serum-containing medium, that more than 10% still express the hematopoietic markers. Another tissue that we try to isolate cells from is the Wharton jelly. This work was done in collaboration with Professor Mark Weiss from Kansas State University, and the study was done in comparison to serum-containing medium that contains, besides serum, also hormones and growth factors. You can see in this slide the results for the isolation of cells from 4 cords, and you can see that in each cord the use of MSC NutriStem, and in this case together with 2% human serum for the isolation step, and you can see higher number of cells in case of MSC NutriStem in comparison to the serum-containing medium in each and every cord. Another tissue that we try to isolate cells from is the adipose tissue, and you can see P0, the isolation step, together with 2% human AB serum, and then P1, and P2 that were done without any addition of serum, just the use of MSC NutriStem together with MSC Attachment Solution. And when we check for the expression of MSC markers, you can see that around 99% of the cells express the MSC markers, and only low number of cells still expressing the hematopoietic markers. Another tissue is the placenta. We try to isolate cells from the placenta, and in this case also without any supplementation with human AB serum. And you can see the numbers in this slide for P0 and P1, and you can see that you can get almost 3 times number of cells using MSC NutriStem and MSC Attachment Solution in comparison to the serum-containing medium. And when we check for the expression of MSC markers, around 98% of the cells express the MSC marker. So, if I would like to summarize the isolation step, we may say that MSC NutriStem and MSC Attachment Solution achieve MSC culture with high number of viable cells, high level of purity, and classical profile of MSC markers. If we try to isolate cells from bone marrow or from the placenta, we don't have to use any human AB serum in the isolation step. On the other hand, if we try to isolate cells from adipose tissue or from Wharton jelly, it is recommended to use 2% human serum for the isolation step P0 only. So we isolate the cells, and now we want to expand the cells in vitro to have a high number of cells available. We did an extensive performance validation for MSC NutriStem and MSC Attachment Solution used for the extension of human mesenchymal stem cells. And this is with regard to the proliferation rate of the cells, mesenchymal stem cell characteristics like fibroblast-like cell morphology, multilineage differentiation, cell renewal potential, and the expression of MSC markers. We also tested the suitability of the system for the extension of human mesenchymal stem cells from a variety of tissues, and we also checked the karyotype of the cells after extension in vitro. So first, the cell morphology. You can see here, when we use the MSC NutriStem and MSC Attachment Solution, you can see typical fibroblast-like cell morphology in comparison to other commercially available media. In this case, you can see some sort of differentiation in the cell, and in this case, you can see low growth rate of the cell. Here you can see the trilinear differentiation of the cells, in this case cells from bone marrow, and in this case cells from adipose tissue. The cells were expanded in vitro using MSC NutriStem and MSC Attachment Solution, and you can see that these cells still maintain multilinear differentiation potential into adipocytes, osteoblasts, and chondrocytes. We also tested the self-renewal potential of the cells by using colony-forming unit fibroblast assays, CFU-F assays, and you can see in this case cells from bone marrow and cells from adipose tissue, and we can get nice colonies that actually originated from a single cell. We also tested the CFU-F assay using cells from the Wharton jelly, and in this case we compare it to serum-containing medium, the white medium, and we use various concentrations of cells. And you can see that you can get more or less the same result for the CFU-F assay in comparison to the serum-containing medium. We did also a study on immunophenotyping of human mesenchymal stem cells from various sources. In this case, cells from adipose tissue. We see that cells in various concentrations, starting from 1,000 cells per centimeter square up to 6,000 cells per centimeter square. These cells were passed for 3 times in vitro, and then we did the immunophenotyping, and as you can see, more than 99% of the cells express the MSC markers in each and every case. On the other hand, only low number of cells still expressing the hematopoietic markers. As for karyotypic stability, you can see here cells from bone marrow and cells from adipose tissue that were cultured using MSC NutriStem and MSC Attachment Solution for 4 passages, and then we tested the cells we did a karyotype for the cells, and as you can see, we got normal karyotype in both cases, in this case male and in this case female. We also did an extensive validation for long-term culture of the cells, and you can see that MSC NutriStem and MSC Attachment Solutions support long-term culture of the cells, and in this case it was done with cells from adipose tissue in comparison to 2 commercially available serum-free media and in comparison to serum-containing medium. And as you can see, MSC NutriStem gave the best results in each and every passage, passage 1, 2, 3, and 4. We did also a long-term culture study using cells from the Wharton jelly. And as you can see here, we tested MSC NutriStem and MSC Attachment Solution in comparison to Stemco and Mesenchymal, as well as in comparison to serum-containing medium, and you can see the results for the 4 passages, passage 1, passage 2, passage 3, and passage 4. And you can see that in each and every passage, MSC NutriStem and MSC Attachment Solution actually yield the best results. And this is the average cell count for the 4 passages, and still you can see the MSC NutriStem actually achieved the best result. These data were published in 2012 in the ISCT meeting held in Seattle, USA. So MSC NutriStem and MSC Attachment Solution promote long-term proliferation of human mesenchymal stem cells from a variety of tissues. As you can see here, cells from umbilical cord, cells from adipose tissue, and cells from bone marrow, passage 1, passage 2, and passage 3. And you can see a nice culture of cells with a typical morphology and high density. Besides testing the stability of the basal medium and the supplement mix, we also tested the stability of the complete medium, the basal medium together with the supplement mix. And as you can see here, we tested a complete medium after 30 days in the refrigerator and after 27 days in the refrigerator, and still we got exactly the same growth curve in comparison to a freshly prepared medium. And you can see the numbers here. We got more or less the same number of cells using medium, complete medium, that was stored 30 days in the refrigerator. So we can say that the complete MSC NutriStem is stable for 30 days at 2 to 8 Celsius degrees. So we have the growth medium and we have the attachment factor, but this is not enough. If we would like to offer customers a complete line of products that will be either xeno-free or animal component-free, we have to take care on the dissociation step as well as on the cryopreservation step. So we have the MSC Attachment Solution that I presented before. It is based on human fibronectin, and it is suitable for human mesenchymal stem cells from various tissues. And just before we use, we have to dilute the solution 1 to 100 using Dolbeco's PBS, and then it is ready to use to pre-cold cultureware. And you can see here that the attachment factor, the use of attachment factor is required for human mesenchymal stem cells, as you can see here. But if you do not use any attachment factor, as you can see here, the cells will not attach to the cultureware and will not spread, and actually will not get any expansion of the cells. So the use of attachment factor is required for human mesenchymal stem cells. Another product that we developed is the MSC Freezing Solution. It is a chemically defined, animal component-free and protein-free formulation for optimal cryopreservation of human mesenchymal stem cells. It is complete, ready to use. It is protein-free, animal component-free, and suitable for human mesenchymal stem cells from various tissues. After thawing, you get rapid recovery and high viability, as you can see here, 1.5 hours after thawing, 24 hours after thawing, 72 hours after thawing, you actually get a nice culture of cells ready to passage. And another product that we developed for human mesenchymal stem cells are the Recombinant Trypsin solutions. These solutions are optimized formulations for efficient dissociation of human mesenchymal stem cells. It is animal components-free formulation, and we have two versions. One version is without EDTA, and the other one is with EDTA. So to summarize the isolation and the expansion, we may say human mesenchymal stem cells from various tissues can be efficiently isolated using MSC NutriStem and MSC Attachment Solution. A higher number of human mesenchymal stem cells was obtained after isolation using MSC NutriStem in comparison to FBS-containing medium. Early cultures obtained from primary tissue show significantly reduced hematopoietic cell contamination in comparison to FBS-containing medium. The highest proliferation rate of human mesenchymal stem cell was achieved using MSC NutriStem in comparison to other commercially available serum-free media. MSC NutriStem supports long-term culture of human mesenchymal stem cells from a variety of tissues, and human mesenchymal stem cells cultured in MSC NutriStem maintain the essential MSC characteristics. Another line of products that we developed for human mesenchymal stem cells is the line of products for differentiation of human mesenchymal stem cells. Most of the differentiation media available in the market contain serum. But serum, as it is undefined reagents, may lead to background differentiation. As you can see here, we use a commercially available osteogenic medium, and we differentiate cells from adipose tissue, and you can see that we can get osteoblasts after staining with alizarin red, but we can get also adipocytes in the same culture because of the undefined nature of the solution. So FBS is not the first choice or the optimized reagent for use for differentiation of human mesenchymal stem cells. So we developed several differentiation media that all of them are serum-free and xeno-free. And we have the MSC chondrogenic, composed of two parts. One part is the basal medium, and the other part is the supplement mix. We have the MSC adipogenic, composed of three parts. One part is the basal medium and two supplement mix. And we have the MSC osteogenic, two versions of the media. One version is the usual one, it is ready-to-use medium, and the other one, also ready-to-use medium, that it is a rapid version that actually you can get osteoblast 5 to 7 days from the start of the differentiation. You can see here the use of MSC chondrogenic medium for 21 days, and you can see that during the 21 days, the cells express the chondrocyte markers like agartine and alpha chain of collagen 10. And when we stain the chondrocyte, you can get a nice cartilage tissue, as you can see here, with cells from adipose tissue, cells from bone marrow, and cells from cord tissue. As for the use of MSC adipogenic, you can see here that the use of MSC adipogenic for 14 days yields a nice expression of adipocyte markers, in this case FAP-P4 and PPAR, and in this case cells from bone marrow, and you can see the immunostaining for FAP-P4. And if we stain the cells using oil red O, you can see a clear picture of adipocytes in each case, in this case cells from adipose tissue, cells from bone marrow, and cells from cord tissue. And in the use of MSC osteogenic, you can see a nice expression of osteogenic markers over 10 days of culture, and on the other hand, downregulation of the MSC markers CD105. And when we stain the culture using alizarin red, you can see a clear staining for osteoblasts, in this case cells from bone marrow, and in this case we used cells from cord tissue, and this was done in comparison to a commercially available medium that contains FBS, that you don't see the staining for osteoblasts very clear, as you can see when you use the MSC osteogenic. So we developed a line of products for differentiation of human mesenchymal stem cells from various sources. As you can see here, cells from adipose tissue, cells from bone marrow, cells from cord tissue. They were differentiated into fat, bone, and cartilage. So we had a mission, and the mission was to develop an efficient, versatile, and consistent serum-free culture system for human mesenchymal stem cells towards clinical applications, and we developed this line of products that contain MSC NutriStem xeno-free medium, MSC Attachment Solution, Recombinant Trypsin Solution, Freezing Solution, and the MSC Differentiation media. The developed xeno-free culture system supports the initial isolation, the expansion, and the differentiation of human mesenchymal stem cells from various tissues. So thank you very much. If you require any more information, or if you have any questions concerning the line of products for human mesenchymal stem cells, you may contact us through our website or directly to our email, either my email or the email of our support team. Thank you very much.